Welcome to Study Time, a televised home learning program produced by Rwanda Education Board.
Welcome again, the Senior Fives, but because like we just talked about, when this is a review, even you, the Senior Sixes, or any one person back home that could have interacted with sciences, whether you're in another field, take this chance up and, who knows what happens. So, in today's review section, like we said in the previous one, we are looking at components from the previous sections of Unit 4 about DNA replication. Make copies. But how do you make them? What do you need? What is the actual process that is going on? So we have one component as a multiple choice, as we do have here. And we have some bit of guidance. You just read a question. You may be given more than 1 or 2, 3 answers that look alike. That's why we need to find the best match. Not every answer is wrong, and not every answer is very correct. So we need to look for the best of the bits that is able to fit into the question. There is going to be a space provided so that you're able to match the rate and just move it up and do the writing. 2, some bit of matching scale. Which terminology can go for which scientific description? So everything is provided for. It's now like putting your head together and be able to wrap it up using multiple choice and matching the terminologies together with the descriptions. If it is a base, what is the simplest description of the base? Can I match it out of the rest of the sentences? And lastly, some bit of short answer questions, like 2 or 3 words or 1 or 2 sentences per given description of the question. The question might be lengthy, but requiring just a short response. But before we go to this, I want us to have a recap of the play, which we viewed in the previous. So it gives us a whole idea of the whole process of replication in only 3 minutes. And then we can use that. If we didn't get the time to go and review in the books, then we can base on the video as a backup and be able to do the work. What do you say? So, here we go. We said again in 3D what actually happens.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end.
You should be able to check the lines. You have the 5, you have the 3, and then you have the 5 the other end and the 3. But let's check. You check the helicase enzyme and the way it opens. So you need to remember exactly what we had as the zip. Opening up the jackets, 2 sections, and then the beginner.
This enzyme makes a small piece of RNA called a primer.
So you have the primer as a starter.
Construction of a new strand of DNA. An enzyme called
So the process begins now.
binds to the primer.
Add up.
The new strand of DNA.
Use the enzyme.
Polymerase
Fix the bases as nucleotides according to the complementary base pairing. You remember that? Check. It leads 5 to 3, and we move. a 5 to 3 direction.
The lagging strand cannot be made in this continuous
But you also have this, which cannot move in this direction, like this before.
make this strand in a series of small chunks.
Now, what exactly happens then?
Each fragment is started with an RNA prime.
Have a primer, move this direction,
DNA polymerase then adds a short row of DNA base.
but make it in small sections. Again from 5 to 3, but this time going to the right. But it keeps jumping. So it frog leaps.
Then added further down the lagging strand.
Another one is added.
And the process is repeated again. Once the new DNA has been made,
You have a problem here.
removes all
The primers have to be removed by exonuclease, leaving a gap behind. That has to be sealed again.
gaps that are left behind with DNA.
But we have another problem. Are these slightly connected sections actually put together so tightly, or we need some bit of glue? So the ligase will have to check for all subsections and glue them up. And then here you have a semi-conservative. But how? An old one and a new. I hope that makes it so clear in one short go that we have new strands forming combined with old ones, but I also at the same time the enzymes involved and at what point each enzyme has to come in. Glue sticking the broken fractions along the new sections. So as we've actually been able to check, how do we sum this up? If you watched the clip, but how do you put all this together if one was to tell you, using annotations, describe the process of DNA replication? What exactly would you need? How would your annotation look such that it has captured all the information in one component, at the same time readable, and gives the whole picture of what we just watched? How would we make moving images static and all in one whole piece? That's our annotation. The whole process on one page. And this is what we have. Are we able to focus on it? Can we go by the stages? 1, 2, 3, 4, and then 5. Would we split the stages in 1, 2, 3, 4, and 5 into seconds as the clip was playing? I don't think, yeah, in a way, somehow. So, there you go. We check. Remember, this is a review. So, take a look at it very carefully before we slide it off. Because remember, this is just to help you the activity. So, I still can't go over it all the time. We had the clip. Now, here we go. Helicase working on. Then stabilizing structures, otherwise it would lose it, so it has to keep in one position. The third, the leading strand being formed, and then it keeps. So the process of replication here non-stop. And then you have these fragments being put in this way and then that way. The primers to start that process, but they can't make it so continuous. So what shall the DNA polymerase do? Once these fragments have been done, what about the exonuclease to remove? Choke off these primers. We don't need them. They help to start, but we don't need them at the end of the day. Kick them out, seal off, and then glue our sections to make a complete section like we do have here. Continuous lagging. So, leapfrog as you work, then get back and fix, and then at the same time, move over and make one complete section. Ligase, links, glues up. Finally, are we ready for the work? Multiple choice. Guidance is that for every multiple choice question you're given, listen to me very carefully here, as you at the same time watch this. That you have approximately 1 minute and 5 seconds to think, get the answer out of the many, and fix it. So if it's A, would I have to write my A here? If it's B, would I have to write my B here? 1. The marking goes so easy here, and at the same time, you leave these sections uninterrupted, not interfered with. So your work looks smarter and more perfect, and it is easy to trace the number. Other than circling this, I think I didn't get it right, then you're scribbling, then you want to circle B. There is so much mess here that even when you're to finally get the right answer, it doesn't add up. So space is provided for you to fix your answer. Have you taken a snapshot of that? I think you have. And now we check out what actually we would expect. Nitrogenous bases, adenine, can pair with what? So we need to think about complementary base pairing, A with T and G with C. So what will the answer be here? Remember, this is just guidance. You are the one doing the work. I'm only giving a guidance on how to get the answer. Guidance for number 1 is complementary base pairing. A pairs with T and G pairs with C. So given adenine, then I would look for its complementary base. Remember that. Number 2, toward G. DNA strands run dash in relation to each other. Remember, every time I did this, so you need to look at my hands and then start thinking, which one is smaller, which one is bigger? So if I was to count that this is my sugar, then you would have 1, 2, 3, 4, then 5. So if I flip my hand down, then that would mean that I'm actually having a 1, 2, 3, 4, 5. So you're having anti-parallel. Grow up, climb down. So in the strands, therefore, we don't have it up like this. Remember when we were using the folders, the file folders, the box files, like here and then we arrange them in one direction and then they flip over. So we have going upwards and then going down. So 1, 2, 3, 4, 5 as the number of carbon atoms on the sugar, as well as the last component. And then we again count 1, 2, 3, 4, 5. So you have a 5 up, a 5 down. So 3 up, 3 down. So that's, whoop. Now, look at what we just have here and think about what I actually meant to be able to give you the answer into this. Could it most likely look like A? It's your judgment. Next question. That a nucleotide in DNA is composed of, nucleotides, a long time ago. Molecules, chromosomes. Where did we meet this again? The DNA structure? Were we talking about RNA? Were we talking about DNA? What do we say are the 3 components that make up a nucleotide? So with that, now we come here. 3 components, a sugar, a phosphate, and a base. But any base like sodium hydroxide? Definitely no. So with that guidance, are we able to pick out the answer and fix it here? Twaji, numerical. Between the 2 strands of a DNA segment, the nitrogen bases are held together by DNA. Climb up, go down, sugars, bases, and a phosphate. So is the connection they're asking me about a sugar and a phosphate or a base and another base? And they're saying the 2 strands. Okay. So a segment of the DNA, like a whole zip, and then you cut 1/2. So between one strand and another, remember the ladder. Urwego wuririraho bubaka. Rufitemo uturongo hagati, imitambiko. N'ibindi bintu bishinze bibiri. So the backbone, the bases, and the locking. So in this area where my fingers are interlocking, could that be representing bases? A, T, G, C, and then they lock up? Yeah. So what force holds them together that we need to break and then separate them? You remember the other zip? And then it opens. Then we zip it up again. We said that that machine within the section of your dressing or your bag represents an enzyme that breaks them down, the helicase, and then it breaks down the hydrogen bonds. Could these bonds be the ones that have been putting the bases together to form a section of the DNA molecule? Okay, so I go down and check. Yeah, I think the answer is there, so find it. Number 5, the nitrogen bases pair with bases that are, okay. Question, is my shoe similar to my foot? When I'm going to buy pairs of shoes, do I look for a shoe that is the same size as my foot? Or should be bigger so that my foot can fit in? My left hand and my right hand, are they the same? And if they are, why is this that my digit here is actually not, I don't know how I would even put it, so it even doesn't work. Meaning that, yes, 5 digits, but this face is left, this face is right. So they cannot in any way be the same. They can't be identical. This is not identical to this. Neither is this identical to that. And there is no way my foot is identical to the shoe I dress. So, during pairing, do we just pair with what is available? Yeah, definitely they have to be around, but which type? Complementary, identical, ball, socket. So if this fits here, it doesn't mean that this fist is identical to this round structure. If this is a socket, this is a ball. The ball and socket join. So you lock in, and so they complement. This is complementing. This is complementing. So A complements with what? With T, G with C. So look for the answer. And the third part of the assignment or the activity is matching. The terminologies we have here, 6, 7, 8, 9, 10, 11, 12, 13, 14, and 15. So from 15 to 6, make a subtraction. That's math. How many terminologies do we have? 9? Something like that. So we need to get a terminology and then match it with a description. I'll do only one for you. DNA polymerase. ASE means enzyme. And so we get a pen. I got a red one. I'm not marking. So I check. ASE must remind me of biological molecules under which we have enzymes. And so ASE is a suffix that is added to something. So we're able to know that, oh, ASE will convert a terminology, a process, a substrate into an enzyme. There we go. So we have to recall back and see during that process, what exactly have we been having? Is it a unit? Is it a category of the bases? Is it one of the 2 pyrimidines? Makes up part of the nucleotides? There's nothing that looks like a catalyst for the reaction. So we have to keep going down. Oh, G, an enzyme that opens up the double strand by breaking the hydrogen bonds. But they are saying polymer. So I need to add up. It's not about breaking. Ooh, careful. And then Two areas formed when the double helix separates during DNA replication, a 5-carbon sugar, an enzyme that opens up, an abbreviation for deoxyribose sugar, makes up part of the nucleotide and is made up of one or more. The process of making up the DNA, one of the two pyrimidines. So we need to check and see, when we say DNA polymerase, where do we match? When we say helicase, again, another enzyme, like we said before. So can we move here and get a pen and check if we pick number 10? The easiest way to do it is this, that you would have, therefore, to write your letters here. These are numbers for the terminologies, and these are letters A, B, C, D, E, F, G, H, I, and J. So instead of crisscrossing and moving lines here and there, the work looks messed up in a way. So why not just move a letter? If it is A, move it and write it here. Deoxyribose, deoxy. It's not a ribose, but deoxyribose. So I now check around. Are they referring to DNA? What exactly do we have? So we check. 5-carbon sugar, but even a ribose is a 5-carbon sugar. Do we add in the DNA? Nucleotides, a subunit that makes up a DNA. So we come here, say for 15, and move our A here. So I'll do that for you as you make the others. So if I pick a pen and write my A here, does it match? That the teacher can easily do the marking on this section, and your work looks neat, and you still can make cross-referencing. Do the rest. Short answer. And what do we have here? You answer the question in the spaces provided. So if you take a snapshot and then later in your own time do the writing of the question set, you can always have the answer against the question for easy planning and making your work look smarter, and direct reference between the question and the answer. So they're telling us to, one, to receive full credit. I think there is some bit of harsh kind of conditions. You want a full credit, your answers should be complete. We don't need half-cooked components. So here you have to create a complementary strand for the basis. Remember, again, here is the hint. Do we still remember complementary base pairing? A pairs with T, held by hydrogen bonds, and G pairs with C, held by hydrogen bonds. That a large base pairs with a small one. If I'm given one single large base, am I able to predict and tell the other small one? So like here, if we were to go straight to the answer, you should have finished this by now. Come on. A with T, C, G, GC, all the way through. I'm even stopping here so that you continue the chain. A segment of DNA that is ready to be replicated. Oh, I love this. Show the process that the segment will go through during replication. Remember the sum up after the clip? You took a picture. Remember the other one we had in the previous lesson. So we have this segment as a whole DNA molecule. How do we separate and break these bonds? Open up. How are we going to involve the enzymes? Because you have it here provided for you, so this is a double strand. Now, look here. Are we ready? So, we break the bonds. The zip opens up. Because we have broken the bonds, which enzyme? 2. Which one starts? Do we have a primer? Next, when it comes off, what do we have next? Polymerase, leading strand, lagging strand, leapfrogging. And we go, then come back, then remove the rest of the components. The same exact component we looked at last time. And with that, we need to think about, can we actually do it exactly the way we have this component? Do we do complementary? Which enzymes do we need? What components shall we involve in our answer as we make the description? That I can use these terminologies, fix these terms as descriptions, and write up and form like the other picture we saw. Finally, the last slide. We need to think now, what exactly is happening here? The process of DNA replication is here by diagram. I need to start asking myself, oh, what are these small circles on the sides? What about these small boxes? What do they represent? Ese ni rwa rwego tugeze kubona cyagihe cyangwa iyo rushanyutse rugasepara, can we put it together? But what is each component on this ladder? If we look at this bracket, this is where the tricky part comes in. Be very careful. X has this small square, a sugar here, and a base. How do we call this component? Can we give a name for the whole of this molecule? And then on this molecule, because this is like this. Remember, they are saying this is DNA replication. So I would never make a mistake to say Y is likely to be U, because this is not what? It's not RNA, we are in DNA. The one enzyme involved in DNA replication, and state the type of reaction it catalyzes. Your choice. Suggests why DNA replication is described as semi-conservative. Come on, you guys. We watched the clip. We saw the sum up of the images. Semi-conservative as in be half super glue, give halfway. I mean, don't be so ukagira kore nyine babandi batarekura. Nimubazi? So in semi-conservative, urekuraho gache mukivanga, then we'll have a new copy. And finally, one old strand, one new strand. That's exactly what it means. It's one mark. Don't write a lot. And lastly, you know cell division, the cycle. At what stage do we have this DNA replication occurring? In the middle of cell division or at the very start? Which takes us to interphase. One mark, one word. And there we go. So with that, as you take this picture, I would love to say thank you so much for following up and being able to update yourself on the previous lesson. And I would say that, yeah, we are good to go. Wrap it up this way, do the questions, and thank you so much.